Afternoon, everyone. Thank you so much for joining today for Industry Insights with HDT Bio. Just a quick run through of the agenda. First, we're going to, I'm briefly going to introduce D2R in about one slide, and then we'll lead right into an exciting talk by Jesse Erasmus, who is the Director of Virology at HDT Bio. And then we'll finish the session with a Q&A period. Just before we get going, a quick note on logistics. You can ask questions yourself using the Zoom Q&A function, or if you prefer, by requesting to unmute yourself. The webinar is currently being recorded and will be available on D2R's YouTube channel after the event. Or if you prefer, I can send you a copy of the presentation. Just email me afterwards. So I think most of you are familiar with the D2R initiative, but very briefly, D2R is a provincially and federally backed large initiative that is an inclusive Canadian approach to genomic-based RNA therapeutics. We received $165 million from the Canada First Research Excellence Fund, and our initiative launched a little over a year and a half ago. A 7-year initiative in total, so it will run until at least the end of 2030. We have an ambitious vision to deliver genomic-based RNA therapies to benefit the health of different populations in 3 main disease areas: infectious disease, rare disease, and oncology. So I believe today's talk is going to be really fitting, as it definitely has applications for at least 2 of these priority disease areas. I also want to say that these types of events, these types of seminars where we invite industry to come and present on their priorities and what they're doing in their companies are really important to D2R, because we do have a big focus on commercialization and on collaboration with the private sector. So this is a great opportunity to learn a little bit more about one potential industry partner that is looking for collaborations and has been setting up operations in Quebec as well. So thanks so much, and I'll lend it over to Jesse.
Thank you so much. Okay, thank you, everyone. So today I'll be talking about HDT Bio's technology, especially focusing on a story around how we are enabling high-dose administration of replicon or self-amplifying RNA, specifically for applications in multi-component vaccination or in oncology, where higher doses of these types of modalities may be needed to kind of overcome breaking tolerance to a lot of self-antigens. Before I start, I'll just kind of go ahead and give some acknowledgements to the folks that were involved in a lot of this work. HDT, I think we're around 60 people now, but today I'll be focusing on kind of just some sub-stories around some of the work by these folks, including Amit Kandar, who's our Director of Formulations, Jim Reed's a scientist in our oncology immunology area, Taishi Kimura, who's an innate immunologist, and 2 members of our RNA core, Scott Delbeck and Troy Hinckley. And then also some work from Nikki Warner as a scientist and a virologist in the team. I'm also showing some work from a collaboration with National Institutes of Allergy and Infectious Disease, Rocky Mountain Labs in Montana, Andrea Marzi and Kyle O'Donnell. And of course, just wanted to point out our leadership at HDT, and we're led by Steve Reed, Peter Berglund, Lauren Beck, who together kind of bring quite a bit of experience in starting up companies and developing technologies and advancing them and maturing them. And of course, also, a lot of this work I'll be showing today is supported by the federal government in the USA, including the NIH and Department of Defense and BARDA. So today I'll be kind of breaking up this talk into a couple different sections. First, an overview on RNA vaccines. I'm sure the audience is probably pretty familiar with this, so I'm going to keep that short. And then kind of connect that to RNA viruses and self-amplifying RNA and how those 2 fields are interrelated. And then pivoting and focusing a little bit on RNA formulation. That's kind of the differentiating technology that sets HDT Bio apart from some of the other technologies out there. And then go into talking about increasing dose, how that's a double-edged sword, and then how we apply this technology in infectious diseases and in oncology. And while we don't have any active collaborations in rare diseases, I can say that pretty sure that there'd be many ways to expand the application of this technology into that area. Okay, so I'm sure everyone here is very familiar with this central dogma that DNA encodes for RNA, which encodes for protein. So I'm not going to really belabor this point. And I'll just skip right ahead to this paper that was published in 1990, which I'm sure also everyone is familiar with, the first generation, so the first demonstration that RNA could encode a heterologous protein and could be directly injected into a muscle to produce that protein. So this was the first demonstration, and this was actually performed following naked RNA, so no formulation involved directly into the muscle. And really, the fact that this paper came out in 1990 and we only have products on the market in 2020 really was due to the instability of RNA and relative to DNA, which really hindered the maturation of this technology. So we fast forward to 2020, just pulling a slide from Moderna, one of the companies that really pushed this technology all the way through to a product on the market in 2020. And just to highlight the way that formulation really enabled the maturation of this technology. So Moderna, alongside many other pioneers in the field, developed what are called lipid nanoparticles for delivery of mRNA. And this enabled delivery and protection of RNA in an LNP formulation that could then mediate delivery of that RNA into the cytoplasm of a host cell. And in that cytoplasm, you could get translation of your protein of interest. And of course, there's many other ways that this can interact now with the immune system, as shown on the right-hand side from Moderna's slide, indicating how this was really utilized to drive robust immune responses in the host. So now switching gears, again, going back to this image on the left of an LNP delivery of RNA and how this relates to virus replication. So this is kind of where I came into the story before I joined HDT, kind of working on alpha virus biology. That's kind of my background. So alpha viruses, these are small positive sense single-stranded RNA viruses. They encapsulate their RNA genome in a proteinaceous core. Let me switch over here to a highlighter, laser pointer. And upon attachment and entry, they actually deliver their RNA into the cytoplasm, much like an LNP. And this RNA encodes two distinct open reading frames, one on the left-hand side that encodes a non-structural protein open reading frame, and then one on the right-hand side that encodes the structural protein open reading frame. Upon initial delivery, this is a positive sense mRNA message, so you get initial translation of this non-structural replicase. And this sets up a replication factory where we now start to transcribe a minus strand template of this RNA genome. And from here, we can replicate, or the virus can replicate a positive sense message. But in addition to doing this, the replicase can now recognize a small subgenomic RNA promoter on this minus strand template and begin to transcribe in molar excess a small mRNA message encoding the structural proteins. Now, this makes sense in the context of this virus life cycle, because every virus particle needs 240 copies of this protein to package a single RNA genome. So it sets up this transcriptional gradient to produce a lot of structural protein. It hijacks a lot of host machinery. This replication complex does a lot more than just replicate RNA. It interacts a lot with the host to antagonize innate immune response and really co-opts cellular machinery to produce as much of this protein as possible. So income replicons, this is essentially where you replace that purple open reading frame with your antigen of interest. This allows us to really take advantage of this transcriptional gradient so that for every single copy of replicon that we can get into a cell, we can amplify and make a lot more copies of that mRNA message encoding your gene of interest. So interestingly enough, this was actually first described 1 year prior to the first demonstration of mRNA delivery in vivo. In 1989, in another paper in Science, they showed that you could encode a heterologous gene of interest using these alpha virus platforms. So just to really highlight that both the mRNA and the self-amplifying mRNA fields kind of emerged in parallel from two very distinct fields. And what I'm highlighting here in red is how this technology was first described. So these RNA replicons were packaged into virus particles. So I'm going to quickly highlight how and differentiate now how these RNAs can be delivered. So back in 1989, this was how it was first described. An mRNA message encoding your replicon and your gene of interest had to be co-transfected into a cell alongside another mRNA message encoding the structural proteins of an alpha virus. And then those proteins were translated, they would make a virus capsid, and they would encapsulate that RNA genome in a virus particle, which was actually called a viral replicon particle, or VRP. And then these VRPs could be purified away from the cells and used as your drug product to deliver the replicon in vivo. Now, what I'm showing here in this figure is some downsides to this approach. One of them being recombination. So this is a rare event that could occur during manufacturing, where now your gene of interest is swapped out with the structural gene. So now you actually are making live virus. So this would occur at very low frequency. There were ways engineered to kind of mitigate this risk, but you could never really fully eliminate this risk. It was always a concern. And of course, because we're using, in this case, a protein shell to deliver RNA, there was a problem of anti-vector immunity. But besides that, and the fact that this required cells to produce, so we're really having to establish robust purification processes to produce this type of product. But besides that, VRPs are the most efficient way to deliver RNA. If you compare the efficiencies of VRP delivery to LNP delivery, it's orders of magnitude more efficient. I think on the orders of 1,000 to 10,000-fold more efficient. So now fast forward to 2012, and Andy Gale, when he was at Novartis, they showed that you could take those same RNA molecules and you could deliver them with lipid nanoparticles. So this completely eliminated the need for cell-based production and really enabled more rapid response and all the upsides that we've seen to RNA vaccines today. The downsides, however, were more limited shelf life, reactogenicity, which I'm going to talk about a lot today in the talk, and of course, reduced transfection efficiency. So now moving forward, I see we have a raised hand. Should I hold or?
I would say that I'll ask Stephanie to perhaps write her question in the Q&A.
Yeah.
We can maybe just remove it there and I'll message her directly.
Okay. Okay, so now I wanted to shift gears a little bit and talk about mRNA and some of the advances there. So I'm sure as we're all familiar, since this was kind of the subject of the Nobel Prize a couple of years ago. Pseudouridine or base modification was put forward as a way to reduce the innate immune response to mRNA. And this was some figures taken from a paper that Moderna had published, where they were replicating some of the work that Katlin Karikó had published in 2005. And here's showing that for an mRNA encoding a reporter protein, human erythropoietin, you could evaluate expression of this protein in the serum of animals when using mRNA based on uridine or pseudouridine. And here you're not seeing a lot of differences in terms of the impact of base modification on protein expression. The biggest variable that seemed to improve expression was this process A versus process B. So the way that they produced or manufactured and purified this mRNA had a bigger impact on protein production compared to the base modification. When we look at type 1 interferon in serum, so this assay is going to come up quite a lot in my talk. This is an assay used to measure type 1 interferon that's produced systemically. It's kind of a marker of reactogenicity. So kind of think of this type 1 interferon response as a reactogenic marker for now. And what you will see here is there was this trend that switching from uridine to pseudouridine, you saw a slight reduction in type 1 interferon induced systemically. But the major impact was actually this process change going from process A to process B that was able to eliminate this type 1 interferon detected in serum. Now, back in 2020, we did try to make replicon or self-amplifying RNA with pseudouridine. And what I'm showing here, this is also a similar study to the figure in panel A here, where we encoded a protein that in this case was a monoclonal antibody, and we could measure and detect this antibody in the serum of mice after intramuscular injection. And so when you look at the replicon data here in pink, we can detect high levels of protein production within 2 to 3 days post intramuscular injection, and this level is sustained for a number of days. We compared that to conventional mRNA. We're seeing much higher levels of protein production as we would expect, given the amplification of this technology. And we also showed here that we could reproduce what Katalin Kariko and Moderna had shown, that you could replace uridine with pseudouridine, and we can still get protein production, albeit it's not any better than the non-modified mRNA. But when we made self-amplifying RNA with pseudouridine, we were unable to get any protein production. So there was something about that particular base modification that was incompatible with the alpha virus backbone. Of course, now today there are other investigators out there who are looking at other base modifications that do seem to be compatible with the self-amplifying backbone. But at this point in time, we had really focused on formulation as a solution to the problem of reactogenicity induced by replicon RNA. So to quickly highlight some of the work that we did, it was really the COVID-19 pandemic that accelerated the development of a technology. We started developing the technology a long time before the pandemic. I think back in 2016 was when we kind of really started working on developing this technology. But in 2020, of course, as we know, the SARS-CoV-2 emerged. We moved pretty quickly to generate data. We were able to get into non-human primates within March. But of course, at this point in time, we had to initiate toxicology studies, whereas Moderna and Pfizer-BioNTech had a pretty robust toxicology data set for their technology, so they were able to move pretty rapidly into human trials. I guess the only claim to fame here is we were able to get a paper out before Moderna. So we did publish in August in collaboration with the University of Washington on our SARS-CoV-2 vaccine approach. And then we did partner up with a company in India who took a license to the technology, and they really accelerated the development of 2 different products, one called Gemcovac 19, the other one called Gemcovac OM, the latter being an Omicron booster. And those vaccines received emergency use approval in June of 2022. And they published some of their results in a Nature Medicine paper that I'm highlighting here for those that want to go back and read. Here I'm doing a meta-analysis, though, of some of the interesting findings that we saw coming out of this clinical development program. So here in this meta-analysis, I'm looking at reactogenicity, so in terms of local adverse events, as well as systemic adverse events. And in the yellow bars, I'm showing those data sets for BioNTech and Arcturus. So BioNTech's conventional mRNA delivered with LNP at 30 micrograms. And Arcturus is an LNP as well, but delivering self-amplifying replicon RNA at 5 micrograms. And you'll see across the board that both vaccine technologies elicited fairly comparable percent of participants with local and systemic adverse events. Now, when we looked at Gemcovac OM and Gemcovac 19, we noticed a much significantly reduced level of these adverse events. And this was not necessarily by design. This was something we just saw emerging in the data that really piqued our interest, and we wanted to find out more. So this vaccine and this data is published, so it can be read more. Just some more details here is that this vaccine was actually developed as a lyophilized drug product presentation, and upon reconstitution with water, could be injected intradermally with the PharmaJet Tropus device. So that's kind of how they moved this product forward in India. So safety and tolerability was demonstrated, and the tolerability was shown to be superior compared to the BioNTech or Arcturus vaccines. And I wanted to mention and make note here that the Arcturus vaccine, which is a rep RNA delivered with LNP, they're delivering this at 5 to 6-fold lower dose than the BioNTech vaccine, yet they're seeing similar adverse events. And keep in mind that the Arcturus vaccine is not base modified, right? At that point in time, I guess all I can say is that these are not base modified replicons, but you still see similar adverse events. Again, keeping that in mind as a kind of a drawback for the self-amplifying technology. Okay, so this is kind of where we wanted to start digging into this difference in reactogenicity that we observed in these clinical data sets. And so here I want to quickly highlight now the major difference between what HDT has developed compared to what some of what many in the field are now moving forward with, which is shown here on the right, called an LNP, or lipid nanoparticle. What we have developed at HDT is a commonly referred to as a cationic emulsion. We call it LION. That's our trademark name for this nanoparticle. It's essentially an oil droplet. It's an oil and water emulsion, where the surface of this nanoparticle is decorated with these cationic lipids called DOTAP. And this imparts a positive charge to these 40 to 60 nanometer nanoparticles. And this actually allows RNA to complex at the nanoparticle surface. This provides quite a lot of flexibility. We can manufacture the LION component completely separately from the RNA, and these formulations can be stored at 2 to 8 or at room temperature. And then the RNAs can be simply mixed either at the bedside or during manufacturing. And we often get asked, well, how does this protect, does this protect RNA from degradation? And this is just that data set showing that we can challenge naked RNA with RNAs, completely degrade the RNA, but when we challenge RNA that's formulated with LION, we can continue or maintain protection of that RNA. We believe this is due to steric hindrance, where perhaps RNases aren't able to really access the ends of the RNA as they're absorbed to the particle surface. But the exact mechanism, we don't know. So just to quickly highlight the two different ways that we can prepare the drug product. So that's the formulated RNA. What I highlighted in the previous slide is this process on the left. So essentially we have LION stored in a fridge and your RNA stored at -80. So this is typically how we would enter into a phase 1 clinical trial. These two components can then be mixed by a simple vial inversion in the pharmacy. So we have pharmacy manual that can describe how this can be mixed. So this really simplifies dose escalation studies. It simplifies the manufacturing, it reduces the cost of manufacturing, but does have some drawbacks in terms of drug product variability. On the right-hand side, we're showing a more recent modification to our manufacturing process where we can now take the LION and RNA drug substances. We've developed a proprietary mixing process to produce this, what we're calling the SVP, or single vial presentation. And then this single vial presentation can be either frozen as a liquid or lyophilized, which is how the Gemcovac OM product was presented to the clinic. Okay, so now this is where Taishi Kimura comes in. So we were trying to understand this difference in reactogenicity between the LNP and LION. And so in this study, again, here, now we're looking at that type 1 interferon response in mice. So this is after an intramuscular injection where we're delivering 10 micrograms of Replicon formulated with LION in yellow or LNP in blue. And then at these early time points, taking blood and measuring type 1 interferon. And what you can see here is that with LNP delivery of Replicon, we do see, as we would expect, an early increase in type 1 interferon that's detected in these animals. And this has been shown by others in the field as well. Now, when we looked at this in the context of LION, we do not detect high levels of type 1 interferon, suggesting that we're at least eliminating some of these systemic adverse cytokines associated with systemic adverse events. So here we're looking at more pro-inflammatory cytokines using a multiplex array. And you can see there are many other of these pro-inflammatory cytokines that were significantly upregulated in the LNP group relative to the LION group in this volcano plot. Additionally, we looked at markers of cardiac damage. So this is measuring cardiac troponin in the serum of animals. And what we see here is a transient, so it's early and transient production or release of cardiac troponin into serum when we deliver rep-RNA with LNP. But when we deliver with LION, we do not detect that marker. And also when we can measure body weight loss, we find that mice that receive rep-RNA with LNP, we see significant weight loss in those animals. But with LION, we do not detect any differences between the naive animals and the LION vaccinated animals. So next we looked at biodistribution. So we had read reports from the IND filing, or the regulatory filings from Moderna and Pfizer. We looked at their biodistribution reports and found that LNPs tend to distribute pretty broadly, even after an intramuscular injection. So we set up this experiment where we labeled LNP with a fluorescently labeled dye, and we can pick up, as reproducing what we kind of read in those reports, LNP can be picked up in the muscle, the draining lymph node, liver, the spleen, and in the lung. However, in the same experiment, in the same group of separate group of animals, when we administered line, also labeled with this lipophilic dye, we only detect signal in the injected muscle and a little bit in the draining lymph node, shown here after long exposure. We've also repeated this experiment using qRT-PCR to detect RNA genome copies, and also HPLC to detect the cationic DOTAP lipid, and it mirrors these results perfectly. So given that lion seems to be maintaining localized biodistribution, so we don't see it just systemically, and we don't see evidence of systemic circulation given in terms of our biodistribution data, but also in terms of our cytokine data. The next thing we wanted to look at was the local response in the muscle. So this is that experiment. Here, the mice receive rep RNA, 10 micrograms formulated with line or LNP, and then 14 hours later, harvesting muscle tissue to run RNA nanostring analysis. And obviously, there's a lot of data. I'm not going to go into all of it, but just to quickly highlight that we see a lot of gene families that are upregulated in mice muscle. These are, each of these columns is an individual mouse. Whereas in the LNP group, we saw lower levels of upregulation, and even some gene families that are downregulated relative to lion. And these were preferentially the families that were upregulated, differentially upregulated in the lion group compared to the LNP group, were those gene families associated with antigen processing, presentation, and B cell signaling. So we tend to set up a robust immune response locally in the muscle environment. Here we're looking at additional cells that kind of infiltrate into that muscle tissue. So these are innate immune cells, CD11b positive cells, macrophages, PDCs, monocyte DCs, and across the board with the lion formulation, we see much higher levels of these cells infiltrating into the muscle tissue. And if you go back and look at the actual composition of lion, it's very similar to an adjuvant called MF59. And the main mechanism of action of MF59 is similar to this. You get a lot of innate immune cells that infiltrate into the injection site. So we think that we're setting up a pretty robust adaptive immune response locally in the muscle tissue. This is a snapshot of what's happening in the muscle. This is 7 days after injection of a rep RNA encoding GFP. You can see that most of the muscle fibers in this tissue section are expressing GFP from the replicon. And then in close proximity to that antigen production, we see a lot of these activated innate immune cells that are coming in and forming almost these like tertiary lymphoid structures as they mount an immune response to that that RNA encoded antigen. Okay, so now switching gears and talking a little bit about how formulation, line or LNP, really, I guess, interact with the host and the immune system and enable differential responses to increasing or escalating doses of replicon RNA. Okay, so to start off, relatively simple here. I just want to quickly highlight here that we looked at 2 different doses, 1 microgram or 10 micrograms of replicon. And in this case, this is an RNA encoding SARS-CoV-2 spike, I believe. And one of the first experiments we did was just ask, okay, if we increase the RNA dose from 1 to 10, what happens to the innate immune response? And so when we look in the context of lion formulation, we see this trend, from in the 2 white bars here, this trend in increasing in type 1 interferon. This is on a log scale. But of course, when we deliver with LNP, we see a much bigger jump in that type 1 interferon response. So obviously, as we would expect, there's a dose-dependent innate immune response to replicon RNA. Then when we look at the adaptive immune response, we see a fairly similar picture emerge between lion and LNP. Both formulations are inducing an antibody response at the 1 microgram dose, and both induce an antibody response at the 10 microgram dose, and there's a dose-dependent increase in the antibody response. So to kind of put this into just a quick cartoon, because this story is going to get a little bit more complicated in the next slide. I'm using pink here to kind of mimic the innate immune response to the RNA vaccine, and then green to indicate the antigen dose, so the amount of protein that's produced from that RNA. So in a monovalent vaccine, you would anticipate as you increase a monovalent construct in dose, you would get a scaled increase in both the innate response as well as the adaptive immune response to that encoded antigen. Now, when we treat these mice with a type 1 interferon alpha receptor blocking antibody, so shown here on the left, we don't change that innate immune response, interestingly. So we can block all type 1 interferon alpha receptors in that mouse by administering a monoclonal antibody. We still detect the same levels of type 1 interferon in these mice. Interestingly, though, when we look at the adaptive immune response now, we see a huge, significant increase in the adaptive immune response just by blocking type 1 interferon alpha receptor signaling. The LNP group was a little more variable, so we're not entirely sure as to why we don't see as tight responses in that group, but there certainly is a trend towards a higher antibody response when we block type 1 interferon. So we think that this is likely due to the changing antigen dose. So this is another experiment we performed a while back, again, looking at protein production from self-amplifying RNA. When we treat mice with that same type 1 interferon alpha receptor blocking antibody, we see significant increase in that protein production in these animals. So it definitely makes sense that as we block type 1 interferon signaling, we can get a more robust adaptive immune response because we're probably producing a lot more protein. So again, showing here in the context of this cartoon, as we kind of restrict that pink box, we reduce that innate response, the protein antigen load kind of increases as a result. Okay, so now what happens if we now incorporate multi-component, multi-antigen, multivalent vaccines into the story? So here we're maintaining a fixed monovalent vaccine dose, but when you add up the total RNA dose delivered across those 4 antigens, you're getting a much larger innate immune response relative to your monovalent antigen dose. So how will this impact both the innate and adaptive immune responses in vivo is the next question. But before we go there, I just wanted to quickly give some background on some of the model antigens that we used to test this, to ask this question. So this is a study where we developed a vaccine for a non-enveloped virus called enterovirus D68. I'm not going to go into a lot of the background of this virus, but it's a respiratory virus, causes acute flaccid myelitis in children, and is an ongoing public health threat. This is a non-enveloped virus, so prior to us publishing this work, there haven't been any papers published on how you would make an RNA vaccine for a non-enveloped virus. These antigens are produced in the cytoplasm, and the way that they're released from the cell is kind of an enigma, so we weren't sure if this was going to work. But in this case, EVD68 encodes these 4 structural proteins as a polyprotein, and it requires in trans this protease from its genome to come in and actually cleave these polyproteins into subunits that form these protomers, and then you get 60 of these protomers that assemble into a virus-like particle. So to produce these virus-like particles, we had to actually co-express that structural protein with a protease component of the vaccine, so a non-structural component co-expressed with a structural component. We used an internal ribosomal entry site to do this. And we just show here with our control, where we deleted the 3CD component, that we don't actually get the correct processing of VP1 unless we co-express 3CD. So this kind of indicated that co-expression strategy was required to produce the right conformation of VP1. So using that vaccine as our model here, we wanted to administer an EVD68 monovalent vaccine at 3.3 micrograms into one group of mice, or into another group of mice, administer that same monovalent vaccine dose, but combine it with 2 other irrelevant antigens, in this case, SARS-CoV-2 spike and flu HA, so that the total RNA dose added up to 10 micrograms. And then with those 2 vaccines, we're now formulating them either with lion or with LNP. And here we're administering a vaccine on day 0 and day 28. And we take an early bleed to look at that early innate immune response, and then later bleeds to look at the adaptive immune response. With EVD68, we were able to set up neutralizing antibody assays. We also have a challenge model, so we could challenge these animals intranasally with virus, and we can measure viral load in their lungs. So beginning with the innate immune response at the early time points, here you can see when we compare the monovalent vaccine to the trivalent vaccine. With Lion, we do again see this trend that we get increase in type 1 interferon, but it's not significant. And then in the LNP group, we see a significant increase in the type 1 interferon. That's dose dependent, kind of like what we showed before. And now when we look at the adaptive immune response, this is neutralizing antibody titers between Lion and LNP. So in this graph here, I'm just showing the monovalent vaccine group. At post-prime, there's no real difference between Lion and LNP in terms of the neutralizing response. Interestingly, post-boost, the responses seems to go down in the LNP group. I'm not entirely sure why that's happening. But in the trivalent group, we completely lose neutralizing antibody responses in the LNP formulated group, whereas within the Lion formulated group, we maintain neutralizing responses, although they are more variable and trending lower compared to the monovalent group. So of course, now when we challenge these animals with EVD68, it's only the Lion formulated trivalent vaccine group that was able to significantly reduce viral loads in the lungs of these animals compared to the LNP group, which was not able to reduce viral loads compared to the negative control. So to now put this in the context of that cartoon, when we were delivering a trivalent vaccine with Lion, where the innate immune response is fairly minimal, systemic innate immune response. So each component is contributing minimally to that systemic innate immune activation. The sum of that innate immune response is likely minimal, or it's not enough to really inhibit the production of the antigen dose. Whereas in the context of LNP, each antigen component is contributing significantly to that systemic innate response. And in that scenario, that sum of the innate immune responses is really at that point at a level where we can now start to inhibit antigen production and adaptive immune response. Again, going back to the EVD68 story really briefly, this virus exists in multiple different genotypes, and we didn't really know the antigenic diversity. We only knew about the genotype diversity. So we used our vaccine platform to generate an antigenic map of this virus, and we were able to show that there was quite a lot of antigenic diversity. So this is now looking at each of the 6 vaccines and their responses against each of the 6 viruses. But based on this data, we identified that we would need to combine vaccines against the A1, B2, and C clades of this virus to really cover the entire species in a trivalent vaccine. So we took that composition, the A1, B1, and C vaccine, and we added 2 more just to really push the envelope here and increase the RNA dose to 100 micrograms. So in a pentavalent vaccine composition here. And we went into non-human primates to really see if we could reproduce what we saw in mice. So this is a study where this pentavalent 100 microgram dose vaccine is delivered with Lion or LNP into non-human primates. And we have a very similar study design, prime boost, early time point for innate response, late time point for adaptive immune response. Starting again with the innate response, we do see really high levels of systemic type 1 interferon in the LNP group. Again, this is 100 micrograms. Recall going back to the Arcturus data, 5 micrograms was really the tolerable dose that Arcturus landed on for their rep-RNA LNP vaccine. Here we're going into monkeys with 100 micrograms with LNP, and you can see perhaps why higher doses when you deliver with LNP are not tolerable. With Lion, however, we do see significant reduction in that type 1 interferon response. We also looked at nanostring in peripheral blood mononuclear cells, and in red are the cytokines that were significantly upregulated compared to the pre-immune time point in each animal. And so with LNP, we're seeing a lot of these pro-inflammatory cytokines present in systemic circulation. With Lion, only 2 of these gene transcripts showed up in this analysis. I'll highlight here IL-1RA and CXCL10 that showed up in the LNP analysis. Those were implicated in this paper to be positively associated with vaccine-associated myocarditis. So we do think that a lot of these pro-inflammatory cytokines are playing a role in the pathogenesis of myocarditis that occurs in a small proportion of individuals that receive LNP vaccines. With lion rep-RNA, we saw minimal upregulation of those systemic cytokine gene transcripts. And now when we look at the adaptive immune response in this study, we see what we saw in the mouse study, where the LNP vaccine is not inducing any detectable neutralizing antibody responses. I can say that we did confirm that the monovalents, as you saw in the mouse study, do induce a neutralizing antibody response. So the vaccine is active as a monovalent vaccine, but in the trivalent composition, where we have this robust innate immune response, we think that is what is driving down the neutralizing antibody response in the LNP composition. With lion, we were able to detect this genotype-specific neutralizing antibody responses. We, of course, think in this group here, the neutralizing responses were suboptimal. We probably need to play with the ratios to balance out the neutralizing antibody responses here. But the main message here is that we could drive neutralizing responses to all three components in that pentavalent vaccine. And when we do a correlation analysis between the type 1 interferon response in each animal and that animal's neutralizing antibody titer, we do see a strong negative correlation, kind of supporting our hypothesis that this is due to some competition between the innate immune response and the adaptive immune response. So in this case, LNP, we believe, is not compatible, at least in a multivalent enterovirus self-amplifying RNA application. Okay, and then finally, just quickly highlight this work where we wanted to actually compare now self-amplifying replicon RNA, our technology, to a gold standard live attenuated vaccine platform. So this one is VSV, or vesicular stomatitis virus, which was recently licensed for use as a single dose Ebola virus vaccine in several countries. So we wanted to know if our platform could induce single dose protective immunity like VSV. So here we collaborated with Andrea Marzi's group at NIAID to develop a Sudan Ebola virus vaccine and compare it to Andrea's VSV version of that vaccine. And just really quickly getting into the data here without going into too much background, what I'll show here is that VSV Sudan, so that's our gold standard live attenuated single dose vaccine, is in black. We did, interestingly, one animal, I think, succumbed to infection here, which I believe is not typical. But at least for the lion Sudan in red, we had 100% survival. We also included a combination vaccine, so a lion rep-RNA encoding the Ebola glycoprotein, which is antigenically distinct from Sudan Ebola virus. The monovalent Ebola virus vaccine did protect 50% of the animals. And also when we combined it with Sudan in blue, we got 100% protection. So no antigenic competition observed in that bivalent composition. And then when we look at weight loss and body temperature, we see a similar story where the red line is really, the lion Sudan or the blue line, the combination vaccine seems to be driving the best levels of protection in terms of weight loss and body temperature. When we look at viral loads, so this on the bottom left is genome copies by qRT-PCR, and the bottom right is infectious virus by TCID50. Interestingly, the VSV group, we'd see some breakthrough viral loads by qRT-PCR. By infectious virus, it's completely sterilizing in terms of viral loads in these tissues. But the lion Sudan or lion combo vaccines were completely, we couldn't detect any virus in any of those tissues post-challenge. And again, this is after single immunization. And yeah, multivalent immunization did not reduce that protective efficacy. And then finally, there were some really distinct humoral responses that I thought would be interesting to highlight here. First off, just looking at the top panel, and there's a lot of different panels there, different colors and bar charts. The main thing to highlight here, though, is that there's very comparable binding antibody responses between the vaccine groups, very comparable neutralizing antibody responses between the vaccine groups. So in terms of systemic binding and neutralizing antibodies, pretty comparable between platforms. But when we looked at ADCD, ADCP, so complement deposition, cellular phagocytosis, and neutrophil phagocytosis, we started to see a pretty different picture emerging. VSV seems to induce much better complement deposition in terms of the functional profile of the antibody response. But Lion, at least at this early time point, this was pre-challenge and this is 6 days post-challenge. Lion seems to be inducing much better cellular phagocytosis and neutrophil phagocytosis. So a very different functional profile of the antibody seems to be induced by the platform. And one of the next questions that we'll be following up with is, could these differences account for the enhanced protection provided by Lion? Okay, and then finally, I'll just quickly switch gears and talk a little bit about one of the programs that we have in oncology. I am not an oncologist, so please forgive me if I say anything incorrect here. But we do have a program called HDT 501 that's designed for prevention of brain metastases in breast cancer patients. And so here I'm just showing a generic figure of HER2, the current therapies available for HER2 positive breast cancers. And HER2 being a receptor on the surface of these cells. And as you have abnormal HER2 signaling, you can get tumor formation. And there is a therapy available on the market today, Herceptin or trastuzumab, that recognizes the HER2 receptor. And speaking of ADCP, ADNP, not only does it bind to this receptor and prevent signaling, it also can lead to destruction of the HER2 positive cells via those ADCP and ADNP mechanisms. But this antibody cannot cross the blood-brain barrier. Therefore, if any of these cells do make it into the brain, this available therapy today cannot really treat these. And so you do have this population of patients emerging that cannot be treated by this particular product. So can our platform induce an anti-tumor T cell response in the periphery that can really then, those T cells can then cross the blood-brain barrier and kind of prevent brain or treat brain metastases? And so what we would like to do is target the same exact protein, the HER2 new protein. We're actually going after the intracellular domain to drive a robust CD8 T cell response to that same protein. And that's a clinically validated target. So to do this work, we're working with Nora Desis as well, who has in this paper shown that a DNA vaccine encoding the intracellular domain of HER2 or ERBB2 does seem to have some signal that it could work. So here is their construct. It's a DNA encoding the HER2 intracellular domain shown here in green. And what they do is administer this intradermally with 100 micrograms of recombinant GM-CSF as an adjuvant. And then just one figure out of this paper showing that there was this potential dose-dependent improvement in overall survival with this particular antigen adjuvant combination. So we would like to know if our platform could potentially enhance this observed effect. So of course, to test this, we've gone back into mice and we've compared Nora Desis's pDNA vaccine combined with GM-CSF in mice to our rep-RNA delivered intradermally with or without an adjuvant, or into the muscle with or without an adjuvant. And what we're showing here are the T cell responses in the spleen of these animals, suggesting that we can drive more robust, at least splenic T cells that are HER2 ICD reactive. And that intramuscular seems to be driving much better T cell responses than intradermal, at least for our platform. We're seeing this reproduce over and over again for other antigens. And the other take home here is we are working on ways to potentially adjuvant the T cell response to our platform further, but we're not seeing any significant enhancement here, but there are trends that we could potentially adjuvant these responses. Okay, so with that, I'll just conclude to say that the current iterations of lipid nanoparticle formulations, they do enable robust delivery of mRNA and rep-RNA. However, I think that technology is currently operating at or near the tolerable dose. You'll notice that all of the doses that were kind of moved forward at licensure for the Moderna, for the Arcturus, for any of these vaccines, were the maximum tolerable doses identified in their phase 1 clinical trials. Except with the exception of, I think, Pfizer, they went down to 30 micrograms and they went a more conservative route. So there is, they're already operating at the tolerable dose ceiling. If you want to add more antigens, add more RNA, it's going to be complicated to do so without taking a hit in terms of safety and tolerability. Rep-RNA can provide dose-sparing effect, and that's how most people are kind of pitching the technology these days. But as you have also seen, that comes at the expense of the tolerable dose ceiling. So you have a dose-sparing effect, but your tolerable dose ceiling is also reduced. So it's not really solving the problem of how you're going to go after multi-antigens or less immunogenic antigens. So that's what we propose to come in and do. We can raise that tolerable dose ceiling, which is going to be required. And we think we do this by lowering the systemic innate reactivity and by localizing that biodistribution. And I'm hoping, convinced you that that the systemic innate response really does contribute to reactogenicity as well as reduced immunogenicity. And I think that second point is, I guess, maybe controversial. I know there's a lot in the field that say you need a robust systemic innate response to have a robust adaptive immune response. But I'm hoping that I've shown you today that it can actually be counterproductive, especially in the context of self-amplifying RNA vaccines. We've also shown that our platform can demonstrate comparable potency to a live attenuated vaccine platform, where we demonstrated single dose efficacy against Sudan Ebola virus challenge. And we've now also shown that we can demonstrate improved T cell responses to a breast cancer antigen compared to a clinically validated DNA vaccine. And with that, I'll end by just saying we have a lot of programs ongoing in infectious disease. So this is just a highlight of some of the different programs we have covering prototype viruses across different viral families. I'm actually here in town today for the American Society for Virology. So we have a lot of, I'm looking to collaborate and expand there. And of course, very interested in talking with anyone in the oncology or rare disease spaces as well. Thank you.
Thank you so much, Jesse. We have a few minutes left for questions. The first one, how does Lion uptake rate compare to that of LNPs across various cell lines? And how can you tailor Lion to target certain organs?
Okay, great question. So as far as the rate of uptake comparison studies, I don't think we've done those studies exactly, but we have compared just general transfection efficiency between LNP and Lion in vitro. And we do see better transfection in vitro with Lion, but I always, say that we don't read too much into the in vitro data. The way that these formulations work in a dish is completely different to the way that they work in vivo. And I think everyone now knows that a lot with LNPs, they depend on interaction with host serum proteins, and then that complex, that corona drives tropism and biodistribution. So what we're doing in vitro is not really indicative of what's happening in vivo. As far as, what was the second question again?
The second part was, how can you tailor Lion to target certain organs?
So right now, the way that we believe Lion works is because we complex this positive cationic nanoparticle with a negatively charged RNA molecule, we can vary the ratio. We've actually optimized that ratio so that empirically, selecting a ratio that gives us the best adaptive immune response. But when we look at what that optimized ratio results in, is it's actually a slightly positively charged complex. So there's slightly excess positive charge that's available. And so what happens when you inject into a tissue is you have a lot of negatively charged moieties on the surface of cells like heparin sulfate. And so you get probably very rapid binding to those negatively charged molecules. So if you want to do targeting, tissue targeting, the easiest way to do that right now is direct injection. So if you want to target, the skin, you inject into the skin. If you want to target the muscle, you inject into the muscle. Now, if you want to do a more systemic administration and have it distribute throughout the body and then accumulate in targeted sites, that's a much different problem. We do think we have ways to get there. Amit Kandar, who I mentioned, who's our director of formulations, he's working on a library of Lion compositions and screening them for differential biodistribution. So we are working on ways to obtain compositions with more targeting. But right now, direct injection of your desired target is a solved problem. Yeah.
Thank you. We do have a question here from Aubrey. I've unmuted you, Aubrey, if you want to ask your question. She's still unmuted. Aubrey, we can hear you, I think. In the meantime, we'll go to another question. Let's see, we have a question here. What is the lipid content when you compare lion versus LNP formulations relative to RNA mass?
Oh man, that's a perfect question for Amit.
If you're unsure, we can always get back to the participant afterwards.
Yeah, I can say the molar ratios of the nitrogen to phosphate, nitrogen being the component of the DOTAP lipid, is 15. So we have excess lipid component compared to RNA on a molar ratio basis.
While we're waiting, in case Aubrey wants to speak, I'll ask you a little bit about maybe your existing collaborations with academia, and if you envision potentially working towards collaborations with D2R.
Yeah, so we are, I guess, different from most companies in that we primarily rely on non-dilutive funding, so through federal funding sources. And so we love finding academic partners who we can work together with to draft grant proposals or project proposals. And then we kind of go after that funding together. And then, yeah, essentially we can provide the formulation, the technology. We can also provide the teams that produce the RNAs. We can design RNAs. We can manufacture RNAs. I didn't have time to talk about it, but we've got a whole lot of experience in process development. We've now moved our process into GMP, and we have a clinical trial that's starting. I think we started screening participants last week. So we're moving that process into the clinic. So we have all of the groundwork laid to take a project and translate it into products through our GMP manufacturing partners, through our clinical trial design team. We have a quality and a regulatory team as well. And of course, the expertise to help design antigens together with our collaborators. Or not necessarily just antigens. We can also design, help design your therapeutic proteins that you want to encode. We can also deliver any RNA molecules. I didn't talk about other RNA molecules that we deliver with Lion. So we also deliver innate immune agonists, like TLR3 agonists, RIG-I agonists. And we are working with NCI, another collaborator, to deliver DNA. So think of Lion as kind of like a platform that can be used to deliver a variety of negatively charged molecules, and we can encode any protein. And yeah, happy to collaborate on funding proposals as well.
Wonderful. So what should our researchers do if they're interested in chatting with you a little bit more about potential collaborations?
Feel free to reach out to Janet and me, and we'd be happy to discuss further.
Wonderful. Thank you so much, Jesse, for a really exciting talk. Really a pleasure to have you here today, and hope to continue the discussion soon. Thank you to all our online participants as well and everyone in the room. Have a great day.
Thank you.